Welcome to the Excel cycle. In today's lesson, we will be talking about the interaction between genes. Some terms we will define are incomplete dominance, complete dominance, codominance, haplosufficiency, and haploinsufficiency. Let's begin with complete dominance. This is the simplest form of dominance and means that a single allele will result in the expression of the corresponding phenotype. For our example, let's say that the color red is dominant over the color white in dogs. In a scenario of complete dominance, red, or big A, will always be expressed as long as one copy is present. That means that for both genotypes, homozygous dominant or heterozygous, the red color will be produced. Only in the absence of the dominant allele will the recessive color white appear. Let's again look at our last example, but this time we will illustrate incomplete dominance. In this form of dominance, the dominant allele, or big A, will not fully mask the recessive allele. A homozygous dominant genotype will result in the dominant phenotype red. Homozygous recessive genotypes will result in the recessive phenotype white. It is with the heterozygous individual that we can diagnose incomplete dominance. Here, we see an intermediate phenotype that falls between the two distinct phenotypes of red and white. We can see that the dominant allele is unable to completely mask the recessive allele. This is incomplete dominance. We can think of this intermediate phenotype as a result of different doses of pigmentation. Let's imagine that each big A allele provides 10 doses of the color red. That means a homozygous red dog has 20 total doses. Each little a allele provides no doses of the color red. A heterozygous individual gets one of each allele. Therefore, the heterozygous individual receives 10 doses from the big A allele and 0 from the little a allele. Because of this, the heterozygous dog displays an intermediate phenotype of pink, as the dominant allele was unable to fully mask the recessive allele. This leads to another important concept in genetics, haplosufficiency versus haploinsufficiency. In haplosufficiency, one copy of the wild-type allele provides enough doses for a functional product. Haploinsufficiency is the opposite. One wild-type allele is not enough for a functional product. This can explain dominant versus recessive mutations. Suppose we have a gene that is essential for survival, and that gene requires 5 doses to be functional. If the gene is haplosufficient, one allele will provide enough doses for it to be functional. The second allele may be mutant, but the organism will have sufficient doses from the wild-type allele for the function of the gene. This results in the presence of a recessive mutation, as the mutant allele is present but not expressed. Haploinsufficiency, however, corresponds to a dominant mutation. In this case, one wild-type allele is not enough to produce a functioning product, which means only homozygous individuals may take on the wild-type phenotype. Let's suppose that 8 doses are required to produce an essential human enzyme, and each wild-type allele provides 5 doses. The mutant allele, however, only provides 2 doses. The only way for this organism to have a functioning enzyme is to receive 2 wild-type alleles and get over that 8-dose threshold. The inclusion of even one mutant allele is enough to inhibit the function of this enzyme, meaning the mutation is dominant. Next, let's talk about codominance using human blood groups ABO as an example. Red blood cells, or erythrocytes, show a clear example of codominance. 3 alleles exist that determine different forms of a complex sugar surface molecule called an antigen. The alleles are as follows. A and B are both dominant over the null allele. But when we see A and B together, we see a unique antigen formed, and both alleles are said to be codominant. A 4th blood type arises when an individual is homozygous for the null allele, blood type O. Codominance is therefore explained as the expression of both alleles in a heterozygote. Human blood groups are frequently used to test understanding of codominance. It's important to note that the I allele is a null allele, producing no surface antigen. Because of this, the immune system cannot target these red blood cells and therefore can be donated to all. Let's summarize today's lesson. Complete dominance is when the presence of one dominant allele will result in the dominant phenotype. Incomplete dominance is when the presence of one dominant allele cannot fully mask the recessive allele, produces an intermediate phenotype. and codominance has both alleles expressed in the heterozygote. Thank you for following along with this lesson of the Excel cycle. Be sure to like and subscribe, as well as comment suggestions for future videos below.